Hello students, it is a Sunday afternoon, and since I am a kind and merciful ruler, I am spending my Sunday afternoon teaching you about the cell cycle and all the different parts so you can pass your quiz this week. So the first part of the cell cycle that we like to talk about is interphase. Interphase is the growth part of the cell cycle. Mitosis is the division part of the cell cycle. And during interphase, our cell looks like how we normally imagined a cell to look like. We have a nucleus and a nucleolus and DNA in spaghetti form, which is called chromatin, and we have an ER right outside the nucleus, and some of it's rough, and we have a mitochondria over here, and we have a Golgi body up here, and we have a lysosome, and we have ribosomes, all the things that our cell needs to do its normal life functions. So the three parts of interphase. First one is G1. That's when the cell does all the things that it needs to do, normal life functions. That's when the cell eats its food, and it uses the energy, and it does digestion, and it produces waste, and it grows and develops and does all the things that a cell needs to do. After G1, we have S phase. S phase is the synthesis phase, and what happens in S phase is we replicate our DNA, and we also produce those nifty little things called histone proteins, which act like spools for our replicated DNA to coil around. They need to take up less space because when you replicate your DNA, you have twice as much in the same volume. It wraps around those histone proteins so it doesn't take up as much space. After S phase, we have phase G2, which is when the cell gets ready for division. And so all of our other organelles are replicated, and we kind of just get ready to divide sooner or later. These phases each last a different amount of time. G1 is about 11 hours. G2 is about S is about 7 hours. G2 is variable. We don't have a time for that just because it kind of depends on what kind of cell you are. If you have a lot of mitochondria, it might take up more time. If you're just a simple cell, then it might take less time. So we don't really have a good indicator for how long G2 lasts. So that's interphase. Take a look at it. Normal function in G1, replicate DNA in S phase, organelles replicate, didn't write that, in G2, and that's interphase. Then after interphase, we get into mitosis. Mitosis is the division part, and mitosis is split up into 4 parts. The first part of mitosis that we talk about is called prophase. You can think prophase is primary. So during prophase, we have 3 really main big things that happen. Our cell looks a little bit different. We have replicated all of our stuff. The first big thing that happens is that membrane that's on the outside of the nucleus starts to disappear. It starts to dissolve. We start to get rid of it because our DNA needs to come out. And that nuclear membrane needs to get out of the way so the DNA can come out. One of the other important things that happens during prophase, is that our chromatin DNA coils up into chromosomes. It's been replicated. It got replicated in S phase, and now we have these nifty little chromosomes coming. So they're all zigzaggy like that because that DNA has coiled up around the histone proteins to get ready for division. So we get rid of that nuclear membrane. And we also get rid of the nucleolus, which was present during interphase and is no longer present. We coil up into chromosomes. And then the last thing that happens is that we have these organelles called centrioles, which migrate to opposite poles of the cell. Just like planet Earth has poles, a cell has poles. These centrioles are responsible for helping us split our chromosomes apart during division. And so I'll write over here. We have DNA turns into chromosomes. I hope this is big enough. We have nuclear membrane disappears. And then we have centrioles migrate. And that's what happens in prophase. You can't really read that, sorry. You can hear what I'm saying. That's good enough. And so, after prophase, we move on to what we call metaphase. Metaphase, I will call the middle phase, because that's where our chromosomes go. So during prophase, we had our DNA coil up into chromosomes, and we had these centrioles move to opposite ends. And now during metaphase is when our chromosomes do the movement. And so our chromosomes during metaphase line up at the center of the cell. The center of the cell is called the equator. Just like we have poles of a cell, we have poles of an equator as well. The centromeres are joined in the middle by this little round disc. That disc is called a centromere. And that centromere is important because when we are getting ready to split, we have these nifty little things that are called spindle fibers. And those spindle fibers get to attach right at that centromere so it doesn't damage any of the actual DNA. So it attaches at these little discs called centromeres. And so those little stringy things coming out of the centrioles are called spindle fibers. They attach to the centromere disc in the middle because eventually those chromosomes will be pulling apart from each other. And so we have them lined up at the equator. And we have the spindle fibers coming out. That's metaphase. Metaphase means middle, and that's where our chromosomes are. They're lined up in the middle or the equator of the cell. Then after metaphase, we have the phase that's called anaphase. Anaphase is interesting because our cell, during anaphase, one of the big things that happens is that it elongates. Instead of being a circle, it's more like an oval now. And so our chromosomes were just lined up at the middle, and now they've started to come apart. So when you cut those X's in half, you have these little greater than, less than looking things. And they each have their centromeres. And then those centromeres are attached by a spindle fiber to each centriole at opposite sides of the cell. Anaphase, I didn't write this. We're going to call it the apart phase. So our chromosomes that were lined up in the center have now been pulled apart. When the spindle fibers shorten, it breaks that centromere and it pulls the things away from each other. These spindle fibers are shortening, and when they're shortening, they're pulling the chromosomes to each opposite side of the cell, because eventually this is going to pinch into two separate cells, and we need half the chromosomes on this side and we need half the chromosomes on that side. And so that's what anaphase happens. We have the cell elongates like a football. And then we have the centromere splits. And then we have the spindle shorten and move the chromatids. So since we have broken that X in half, these things are now called sister chromatids because we cut the X in half, and when we cut the X in half, we cut the centromere in half. Eventually, that centromere will repair, become a full circle again, and when that centromere is a full circle instead of just being a half circle, these things get called chromosomes because they are by themselves and they have a full centromere. When it has a full centromere, it's a chromosome. When it has a partial centromere, it's a sister chromatid. Sister chromatids are identical to each other. So now they're chromosomes, they're just unreplicated. It looks like an X because it's been replicated. You have equal halves on either side, but those things have been cut in half and you just have the original on each side. So anaphase, our cell elongates, the centromere splits, the spindle shorten and move the chromatids to either side, and then those centromeres repair, and they're once again called chromosomes. And so after anaphase, we have the last part, which is called telophase. And we'll remember telophase because it's two cells. the phase where we have two cells. So in telophase, our cells start to kind of split in half. You can see that little peanut in the center means that they're about to split from each other, and then the process that causes them to ultimately split all the way is called cytokinesis. But first things first, we get our nuclear membrane and our nucleolus back, and all my chromosomes are sitting in there. All my chromosomes are just hanging out in that nucleus that has reappeared. The chromosomes will eventually uncoil, so they will undo the little wrap that they've had, and they will go back into chromatin form, which is spaghetti form. Okay, and then after that, we get rid of our spindle fibers. We don't need them anymore, so those are gone. You notice you don't have any dotted lines in there. And then we start the process of cytokinesis. Cytokinesis is splitting the cytoplasm to completely break these two cells into separate pieces. This cell that I wrote is an animal cell. In animal cells, we have this thing called a cleavage furrow. And that means that it's starting from the outside and pinching inwards. If I had a plant cell, we'd have a slightly different structure. That slightly different structure would start in the middle, and it's called a cell plate. So the cell plate starts in the middle and kind of expands one way and expands the other way and expands and expands and expands until it stretches the length of the entire cell. And when that cell plate is all the way formed, those cells can come apart from each other and separate into two original cells. And so the things that happen here, we have our nuclear membrane come back. Our chromosomes uncoil. And then we have cytokinesis happen. In addition to getting rid of those spindle fibers. We cannot enter interphase until we have completed the cytokinesis part. So until this cleavage furrow pinches all the way inward and I have two separate cells. And until this cell plate stretches from one end of the cell to the other, then cytokinesis is finished. Once we finish cytokinesis, then we go back into interphase and start the whole process all over again, doing our normal life functions. Eventually the DNA will replicate again. Eventually the organelles will replicate again. And this is our cell cycle. So, in case you've forgotten one more time, I'm going to show you what it looks like. We have interphase is what it looks like when we're a choir girl. Notice you can't see my fingertips. Okay, my fingertips are my DNA. And we cannot see our DNA during interphase because I have a nuclear membrane in the way. We can't see them until that nuclear membrane gets out of the way. And so once that membrane gets out of the way, we're in prophase. I can see my fingertips now. I can see the DNA because that nuclear membrane is getting taken away. It's disappearing. My DNA is coiling into regular old chromosomes, and so that's kind of what my little long fingers look like, are our stranded chromosomes. Metaphase, the chromosomes line up in the middle of the cell, also known as the equator. You can see them because my fingertips are my DNA, and you can see them lined up in the middle. Then we have anaphase. They're coming apart from each other, so you have some going this direction and some going that direction. And then telophase, I have two new cells and two separate membranes. Notice again, you cannot see my fingertips still, because during telophase, the nuclear membrane reappears. And when the nuclear membrane reappears, you can't see inside the nucleus. So we have interphase, prophase, metaphase, anaphase, telophase. Hope this helps. Good luck on your quiz.